I'm Sean Hill, co-director of the Blue Brain Project and head of the Division for Neuroinformatics. I'll be talking today about some of the neuroinformatics issues and challenges in organizing all of the data needed for the data-driven modeling process. So I'll start off taking you through the importance of metadata and provenance and how we use that to build up the knowledge graph. I'll introduce ontologies. I'll give some examples of text mining and data integration from the literature. And I'll also just give a quick overview of the brain atlases and search and interface to our knowledge base. So first of all, the challenge in neuroscience is really that neuroscience data is extremely heterogeneous. Nearly every piece of data is gathered in a different lab and with different protocols and different experimental techniques. And the challenge is how do we take all of this data, which is really ranging from the subcellular level, from gene expression and proteomics, to the cellular level with individual cell electrophysiology and morphology, up to the tissue level and whole brain scale with the new techniques coming in to measure whole brain activity, EEG, behavioral measurements. How do we actually integrate that data? How do we organize it so that we can find the relationships between those different levels and ultimately build models that help us go from gene to behavior? And this is the challenge in neuroscience. And the way that we approach it in Blue Brain is really to establish an iterative cycle of data integration by building models, by building digital reconstructions that help us understand how do the pieces of data actually fit together. And then if we take this step by step by starting from data, available data from the literature, existing models, existing data repositories, we have to organize that data into brain atlases so that we can start to use it to build models, run simulations, and then perform a validation step. And that validation is really central to the process because it's only through validating the model by showing where does it work and where does it not work? Where is it consistent with biological data and where does it fail to recreate the biological phenomena that we know occur? And it's through that process and iteratively performing that process that we improve the models, that we identify new experiments that should be performed, missing data that would be critical to filling in key gaps between the levels, and overall starting to identify the principles by which this whole brain operates across different scales. And the neuroinformatics platform is really the key to supporting that process. It's at the center of the process by enabling the data to be organized, data that may be distributed around the world. And there we've got different components of the neuroinformatics platform. One is the data space, which is really where is the data situated, distributed around the world. We've got data registration, a process by which we annotate the data with the metadata necessary to discover it and to anchor it in atlases. We've got the knowledge graph and search, which I'll talk about, which allows us to actually find and discover data and related data, so any type of data and its properties, as well as atlases, which help us to navigate the data, discover data related to particular brain structures, and also anchor data into a standard space. And finally, we have the knowledge space, which is an online community encyclopedia, which really provides a place where the current state of knowledge can be maintained in terms of an encyclopedic article, but also links to data and data that are either in the knowledge graph that we're talking about or in other curated databases from around the world. So where is the data? And this is actually a massive challenge for neuroscience because most of what's the so-called long-tail data is actually in printed documents or it's unpublished. There are about 100,000 publications each year in neuroscience. But unfortunately, a print publication or a PDF is not a machine accessible way to get data or even to get knowledge. It really requires reading the paper, understanding what is talked about and seeing if you can extract the data from that paper manually. And that's a massive challenge. And it's not an efficient way for the future of sharing data. What we see on this graph is that there is organized big data, and the Allen Institute for Brain Science is probably the best example of generating high quality, large scale data. But as I was saying, most of the data is actually unpublished and dark data, or it's referred to in papers. And there's a big challenge of how do we discover that? How do we find it? How do we organize it? How do we make it accessible? And the way that you do that, you've got to be able to make it possible to combine data. So in order to find the small data and to make it possible to combine it to make big data, is that you've got to make that small data discoverable. You've got to make it machine readable, accessible. You've got to use standardized metadata, standardized data types, data structures, and identify which data has actually been produced using a compatible experimental method or from a compatible experimental context from which data can be compared. And that's a massive challenge. Now, there are different ways of organizing data. One of those is to put them into folders on a hard drive, and this is quite common. When you ask for somebody to share their data, very often it comes into folders, and it's a set of different files, and you navigate the folders to find the pieces of data that you're looking for. Now, a somewhat more sophisticated approach is to actually have searchable metadata that tags this data with properties that are then searchable. And this gives you a lot of flexibility in how you can discover and access the data. So we really think that having metadata annotations for the data files and making that searchable is really key to having a much more flexible way of discovering and accessing the data. So there are three main classes of metadata. There's descriptive metadata, and that's really data that describes the key properties of data for discovery and identification. That includes things like the title, the abstract, the author, maybe keywords that help identify the data set. Structural metadata is information about the structure of the different pieces of data. How are they put together? For example, in a document, how are the pages ordered to form chapters? Administrative data, metadata, is actually that metadata that provides information on how that resource can be accessed, by who, what file type is it. It helps provide information about who can, who should, and what ways in which can that data set actually be accessed. And that's important for things like protecting privacy, or keep knowing when the data set actually hasn't been accessed for a long time and can be archived to tape. But coming out of the laboratory, that's really the point where you want to have the richest metadata annotation. In fact, one of the greatest challenges in current neuroscience is that when you get a data set, it usually doesn't have the metadata that you would need to actually understand the contents. And it would be a lot of work for the experimenter to go back and add that metadata. So we're really advocating that at the point of data capture in the laboratory, that you capture all of the metadata needed. And some of the best practices that we would advocate are using electronic notebooks, use a laboratory information management system, electronically capture the details of the specimen. What species is it? What experimental protocol are you using? What is the data captured from specific experiments? Document the experimental design. That includes having the hypothesis clearly documented, the objectives, the protocols are really critical, and the provenance in terms of which data samples came from which specimens and so on. Use standardized vocabularies when recording metadata values. This is really essential because in the end, when you want to analyze this, when you want to discover it, you want to be as consistent and as rigorous as possible in the way that you annotate the data. And also, finally, curate the data. When you're selecting a key set of data that is used for a publication or is used to be shared with the community, select the high quality data. Make sure that you flag and or remove the data with questionable quality. You, as the experimenter, really are the best person to judge, and the best time to make that decision is when you're very familiar with the data that has been captured from the experiment. One of the additional challenges is, well, what metadata is really needed? How do you find neuroscience data sets if you're talking about any possible type of neuroscience data set? And this is a big challenge, and it still remains to be addressed by the community in terms of setting a common standard for what is the minimal metadata needed to describe a neuroscience data set. Here we've developed an initial proposal. We want to see this developed further in collaboration with the community, but also adopted widely. So our proposal is what we call the Minimal Information for Neuroscience Data Sets, MINDS. And that means what that includes is actually capturing details of the subject of the experiment, the species, the strain, the age, the sex, the methods used, the protocols, the equipment, and the parameters used to perform the experiments, the classification, what is the data category, the data format, the cell type, the brain location, where in the brain in a standard atlas, either in terms of coordinates or in terms of just a standardized name for that brain region, was this data captured? Who are the contributors and what are their affiliations? And ideally, identify those contributors by something like an ORCID, a standard identifier for authors, so that they can get credit for making their data available. And then, importantly, a URL or a persistent identifier, and this is something like a DOI, that allows you to actually go online and directly access the raw data. So that URL is a key part of the Minimal Information for Neuroscience datasets. So in this session, I've talked about how neuroscience data is very heterogeneous and some of the challenges in organizing it. I've described to you what metadata is and how we can use metadata to organize datasets. And I've also described to you MINDS. It's a proposed initial standard for a minimal metadata set for neuroscience data, and we think that's really essential to starting to get the broader community to consistently annotate and organize their data.